This is a production of Cornell University.
Topic today is GM crops as catalysts for global development. And I want to point out to you that GM crops have already played a big part in global agriculture, but that they have a lot more to offer. And if they're handled effectively and safely, can make a big difference to the globe, and particularly to excuse me, developing countries. So those are the main points I want to make. So the actual issues I'm going to address is very briefly, what is a GM crop? Does everyone here know what a GM crop is? Hands up if you do. Oh, I'll put my hand up too. Are GM crops safe? When should one use? Oops, sorry. When should one use GM crops? What has been the impact of GM crops today? And I think many of you will be surprised by the extent of the impact. The enormous potential going well beyond GM crops for global development. And suggesting a strategy that we've been using here at Cornell with a USA funded project on how to do this work on broadening that range in a safe manner. So just for those who are a little bit rusty and haven't been in a biotech class for a day or two, I'm getting, sorry about that. Which way do I go here? It's all counterintuitive. Okay. You see, I'm very hot on technology. I hope you're impressed by it. Genetically modified crops, also known as biotech crops, and genetically engineered crops, GE crops, are developed by the selective addition of a trait from any source. So this is why you hear about tomatoes that might have fish genes in them, which scares the heck out of a lot of people. Why they have bacterial genes in them, etc., etc. I won't go on because if there's any Greenpeace supporters here, you'll probably kill me before I get to the next slide. But any source, but they aren't the main things I'm going to be talking about. Well, the bacterial may be, but sources such as other plants are going to be a major point I'm going to talk about. A lot of people discuss the safety of GM crops. And you'll find it's a little bit like whether we should build a wall between Mexico and America, I mean USA, whether Britain should stay in Brexit or not. There's a lot of opinions, and a lot of the comments are made not on the basis of fact, but on belief. But here are two statements. I always tell my students and the fellows not to use too many words on a slide, so I'm glad that I don't think any are here at the moment. They're all in some international programs event. But the World Health Organization said in 2014, GM foods are currently available on the international market have passed safety assessments and are not likely to present risks for human health. In addition, no effects on human health have been shown as a result of the consumption of such foods by the general public in the countries where they have been approved. Then you've got National Academy of Sciences, 2016. The study committee found no substantiated evidence of a difference in risks to human health between currently commercialized genetically engineered, GE crops in this case, and conventionally bred crops. Nor did it find conclusive cause and effect evidence of environmental problems from the GE crops. So there's a couple of pretty important bodies saying that these crops are safe. What we need to remember is that they need to be kept safe. They need to be all the time, like with pharmaceuticals, etc. There has to be vigilance to make sure they stay safe, both in terms of human health and animal health, and the environment. So when should we use GM crops? I know some fanatics who say, some scientists who say they should be used for everything. They should just take over. There are not very many of those people, but there are some people who believe that. I say, and many people, I was going to say like me, but my wife says there's not too many people like me. I think she means that in a positive. GM crop technology is a tool in the toolbox, not a silver bullet or cure-all, a complement to other approaches, and to use when other approaches are too lengthy, expensive, or unavailable. So don't think you're looking at a sort of fanatic who says all of you should use GM crops all the time. What are the impact of GM crops to date? Well, this is what's happened in terms of global area of biotech crops, or GM crops, or GE crops. From 1996, when there were virtually none grown commercially, to 2017. You see that the total number being grown in 2017 was 189 million hectares. 100 of that 100 million hectares was grown in developing countries, and 89 million in industrialized countries. Which are the main traits, genetic traits that are being commercialized? It's herbicide tolerance is the number one. Insect resistance. And then the two together, what they call stacked traits, to put the two types of resistances together. And in terms of the principal biotech crops, they are cotton, soybean, maize, and canola. And you can see here that there are various amounts of the genetic engineering contributing to the total adoption of those crops. The basic thing is that what I want you to remember is that cotton, soybean, maize, and canola are the main ones that are being commercialized. And that herbicide tolerance, insect resistance, and the combination of those are the main traits to being commercialized so far. So what has been the contribution of these biotech crops to food security, sustainability, and climate change? I won't dwell on any of these, but I just want to give you a picture of a study that has been done by Brooks and Barfoot and published in 2018. These guys have been looking at the impact of biotech crops for years, but this is the most recent thing. Biotech crops have been responsible for increased crop productivity. They've been responsible for providing a better environment, for conserving biodiversity, a point that I'm going to come back to later, reducing CO2 emissions, and helping alleviate poverty and hunger. By the way, if anyone wishes to have a copy of these slides, I'd be happy to send them to you. Perhaps at the end, I'll just put my email address up there again so you can ask for those. Or I can give them all to Martha and she can send them out to people one by one. So that's it so far. A few crops commercialized on a big scale, a few traits. That's it. So what about the rest of the story? What's the next generation of GM crops going to be? Well, it's going to be many, many crop by trait combinations, but here are a few for you to look at. Abiotic stress tolerance, drought, heat, flooding, etc. are going to be addressed and are being addressed. Nutritional enhancement, disease resistance. These are being addressed right now. But, and this is what I find the most exciting thing of all, is that there's a vast range of traits that can be put into crop varieties through genetic engineering. So genomics and bioinformatics are facilitating easier identification, isolation, and transfer of genes for most agriculturally important traits. It's an increased ability to extract the needed genes from plant germplasm collections. The improved use of germplasm collections will strengthen conservation efforts. Now, Martha mentioned that I worked at the International Potato Center in Peru. It's a global operation there. One of its main functions was to maintain and utilize a massive collection of potatoes. Cultivated potatoes, wild potatoes. We had just about every single trait you can imagine within that collection. The downside was that less than 10%, closer perhaps to 7%, depending on who we were writing a report for, that was being utilized to improve crops. Why? Because through regular plant breeding, you had to, well, first of all, identify those accessions, those units, should we say, within the collection that had interesting characteristics such as disease resistance, flood tolerance, etc. Then, through breeding, you had to take those very exciting looking accessions and cross them with the varieties that had everything but that. Then you had to spend several years back crossing to get rid of any undesirable traits that you crossed in through the plant breeding. With GM, of course, as I said earlier on, you can take a particular trait and plug that in to the variety. I'm not saying it's as easy as that. You've got to do all kinds of tests to make sure that plugging in happened right. And there's no deleterious effects of having put that gene into the genome, etc., etc., etc. But GM can speed up the process of plant breeding very dramatically because you're selectively putting in that gene you want. So, not only does that give us the chance to more rapidly develop important new varieties, but it also helps to conserve germplasm. Reason? Even when I was at the Potato Center, it was very, very difficult and increasingly difficult to get the money from donors to maintain the collections we had, even though we had what we modestly dubbed the world potato germplasm collection. And why was that? Because so little of it was actually being used to boost the varieties around the world. What if you saw those plant collections as collections of genes, a field of genes, and you could go in there with molecular biology and pull out the genes that you want and plug those into the commercial varieties or whatever that you want. If you saw things that way, you would increase the chances of people saying, hey, those germplasm collections are now being used to much greater extent. The other thing is, this would help with the perception of many about GM crops. I said at the beginning that fish genes, bacterial genes, etc., are put into the crops. What if the main line in the future was to take plant genes from germplasm collections and plug those into your target varieties? Plant genes into plants. I think that might help with the perception, and perception is an extremely important part of this whole GM crop debate that's going on. I'd be happy to discuss that if there's time afterwards. I'm glad that Susan McCooch is here, because if you don't understand this statement up here, she will explain it to you much better than I could. But back in '97, this was just after I started at the World Bank, the whole place was excited because of an article by Steve Tanksley and Susan McCooch, and I'm not exaggerating, Susan. They said, the tools of genome research may finally unleash the genetic potential of our wild and cultivated germplasm, just what I've been talking about, for the benefit of society. That was a very exciting statement. It's led to all kinds of new thoughts about the area of GM crops and crop science in general. So that was published in Science, and congratulations for doing that work. So we can move beyond a very narrow range of traits, genetic traits. What about the crops themselves? The multinational life sciences companies have focused on internationally traded, big selling crops. I mentioned them earlier on, the soy, the maize, the canola, etc. But many crops have been overlooked, despite their nutritional and economic importance to poor regions. Such orphan crops cover 240 million hectares in developing countries. And there's enormous potential for genetic engineering of those orphan crops to make them more productive, more tolerant to climatic conditions, more resistant to diseases, insects, etc. Back in 2002, we started here at Cornell a project which went under the unlikely name of Agricultural Biotechnology Support Project number 2. I remember describing this to people and saying, what are you working on these days? Well, imagine you're at a party. Well, I work on the Agricultural Biotechnology Support Project number 2. How fascinating. ABSP2 for short. ABSP1 was run by Michigan State. They did a lot of work to help educate people around the world about biotech crops, and supported some good research and so on. But when it came to the next phase, ABSP2, we were asked if we would lead that. And we worked with a whole bunch of institutions, not only in the United States, but in India and Africa, Bangladesh, Philippines. The focus of ABSP2 was to try to develop GM crops in Africa and Asia in accordance with what was in demand there. I remember that I caused quite a stir at USAID when I sent in a proposal which was contributed to by many, many people. And it basically said, we don't know what we're going to do in the first 6 months. You imagine this went really, really well in Washington. I even had a phone call about it, not congratulating me on this brilliance, but what do you mean you don't know what you're going to do? I said, well, there's one thing we did mention. We're going to find out what we're going to do. We're going to get together with appropriate groups, public, private sector, etc., in these various countries who've been asked to work in, ask them what they want. Then we work out whether it's feasible to work on those things. Oh, okay. So we did that, and this project's been going on. Well, it went on for 12 years. Now there's fragments of it splitting off into sub-projects, and ABSP2 itself no longer exists. But it went for 12 years, and I think it totaled about $80 million. I noticed some of you molecular biologists and people, $80 million is just chump change, you know. But at the time, it seemed like a lot of money to me anyway. The entire budget of the potato center was 25 million a year. So you can imagine that getting 80 million was nice. So let's see. As I say, at Cornell University, University, USA-led consortium that complemented Asian and African national efforts to use GM crops safely and effectively. We decided on a product-driven approach, meaning all the activities would be around a product, meaning a particular GM crop. We wanted to boost productivity and sustainability. We wanted to strengthen through the products themselves. We wanted to strengthen national and regional biotech capacity and partnerships, and build a portfolio of success stories about impact in farmers' fields. So we had that aspect, impact assessment, built in right from the start. My American colleagues particularly didn't like this because Sir Arthur Helps was an Englishman, but he said nothing succeeds like success. We wanted to show what was successful in this project and then catalyze more and more success. This is what Ronnie Kaufman elegantly calls our petal diagram. Can you imagine him calling it a petal diagram after the flower's petals? This brilliant piece of strategy put together by myself and all these others working in this project, come up with this fantastic strategy, and he calls it a petal diagram. But anyway, so much for Ronnie, who's not here, so I can insult him as much as I like. But basically, this here was the essence of the strategy we used. And the main thing I want you to realize, if you don't already, is that developing a genetically engineered crop, or GM crop, is not just a matter of going into the lab, using a gene gun or agrobacterium-mediated transfer of genes into a crop and doing selections after that. You have to think about many different things. And we try to harness ideas coming from all angles and put them all into each of the products that we worked on. First of all, it had to be demand-driven. It had to be agreed upon by the countries working on this that this was something they wanted, and it had to be feasible to do it. Had to take a holistic, integrated approach that took into account technology development itself, marketing and distribution, policy, including intellectual property rights and regulatory issues, outreach and communication. Monsanto has had so much trouble with GM crops because they virtually ignored outreach and communication to the public. So the opponents of GM crops could come up with all kinds of things, and Monsanto was not poised well to combat those things. They had not provided public with facts, even though they had masses of facts. When I was a Beltway bandit for 2 years in Washington, consultant, one of my jobs was to review and develop packages, regulatory packages for GM crops. One of the clients was Monsanto. And you'll be amazed by how much detail they go into each time they produce one of these crops in terms of the safety assessments. Sometimes 10 or more years of research goes into that for health, for environment. Then you've got a framework here of consisting of the FDA, USDA, EPA working together in coordinated fashion to assess the safety data that's come out on each of those crops. None of that was communicated to the public by Monsanto. Brilliant science that they did. But having not looked at the outreach and communication very carefully, it was very tough for them. Then, of course, we were doing public-private efforts. We were building capacity within the countries to not only develop, but to assess the safety and efficacy of the crops, and also developing this capacity for product stewardship, looking at the product, the GM crop, after it had been released. We selected several products after discussions with the various nations. Fruit and shoot borer of pest, major pest of eggplant in India, Bangladesh, Philippines. Late blight resistant potato in India, Bangladesh, Indonesia. Disease and nematode resistant bananas in Uganda. I won't go through all of these, but each of those is either commercialized, like the fruit and shoot borer resistant eggplant in Bangladesh, that has already been commercialized and is on the market. And I'll give you a summary of the other things in a minute, but they're either commercialized or on track for that. These products were initiated but discontinued. We thought we'd set ourselves up as just like a corporation. See how things go, something isn't going, that's it. No research for the sake of it. So papaya ring spot, drought and salt tolerance in rice, multiple virus-resistant potato, and tobacco streak virus in groundnut. We stopped each one of those, and stopping one or two of those almost made me cry. By the way, I should point out that the person who's been leading this project extremely effectively for most of its existence is Dr. Frank Szotkowski, who is an adjunct professor in plant breeding and genetics, a very close colleague and friend of mine. And he came in after we had developed this whole concept and got the thing launched and all that. And he was absolutely the right guy to do it, because he used to be a product manager in Syngenta, which is very strong on GM crops. So it was almost perfect. I'll not go into details about why USAID didn't want to continue with me as the director. But anyway, it turned out that they had much more vision than I thought. And Frank came on board, and he was absolutely terrific. And he's not working on it anymore, but he's doing other things all over the place. All right, so we just taking one example of where I think ABSB2 is successful. Look at the eggplant. Now, eggplant, for most of you, is something you have if you're not particularly hungry after you've had a meal. I mean, it's just on the side for most people. But in India, Bangladesh, Philippines, for example, it's a staple food. It's extremely important. It's a crop of great economic and nutritional importance. In India, 470,000 hectares alone are grown, and it's common to all income groups. There was a 50 to 70% yield loss, 50 to 70% yield loss due to the fruit and shoot borer in India alone. Chemical pest control measures were not working, to the extent there was massive overuse of chemicals, major threat to health and environment. There were stories of so much pesticide being around in farms where they could actually afford it, that the kids were drinking it and stirring it with their arms and things like that. Farmers were so worried about the amount of pesticide they were using, they didn't consume their own produce. They would only sell it. And the fruit and shoot borer, like so many insect pests, develops resistance to the chemicals that are being thrown upon it. Also, the increasing cost of chemical pesticide shrunk the farmer's profit. So it was an absolute disaster. We got together a team of globally renowned scientists to look at this situation. We've been told by the countries that they wanted something done about this. This was their top priority. But we wanted to know, was there any alternative to using GM crops? Basically, the answer was no. Conventional breeding was unfeasible. There were no agronomic practices that would cope with this insect. And that if BT eggplant, genetically engineered eggplant, BT I'll come back to in a second, Bacillus thuringiensis eggplant, it was considered by economists to be a viable alternative to the chemicals. I put this up here. My wife is an artist, and I thought you might also appreciate that picture. It's actually of BT toxin, or one of them. Pretty amazing. And all kinds of changes can be made to that molecule to make it effective in different ways. But anyway, getting back to the main point here, the BT gene that was used in this eggplant was Cry1AC, and it is from Bacillus thuringiensis. It produces an insecticidal protein that has a toxic effect on many caterpillars, including fruit and shoot borer. The transgenic eggplants containing this BT came from a company called Mahiko, Maharashtra Hybrid Seed Company. They had already developed some eggplants that were resistant to the fruit and shoot borer, but they did not want to follow through with commercialization, at least not by themselves. So this was a tremendous step forward, that they were willing to donate that to this project, our project, pro bono, free of charge. So we not only had that, but we had the history of BT crops, which was, BT had been used in many different crops around the world for decades and had been highly, highly effective and safe in terms of the environment and health. So the expected impact of BT eggplant in India was there'd be a reduction of crop losses, and we did this with economists and biologists and so on, reduced health and environmental risks, savings in crop production, costs, positive economic impact was estimated $164 million per annum, just in India alone, and provision of food to several million people at affordable prices. The current status of BT eggplant commercialization is that in Bangladesh, as I mentioned, there's already commercialization of this crop, this BT insect-resistant eggplant. Nearly a fifth of Bangladeshi eggplant farmers are cultivating BT eggplant. In the Philippines, it's in the process of being released commercially. Hopefully this year it will happen, but you never know. There can be an intervention at the last minute by the opponents of GM crops with the government, and that can hold up things again. And this is what happened in India. India was meant to be our flagship. This is where this was going to go straight through, and it had all the green lights you can imagine, the various ministries and groups and this and that and the other, and trials had been extremely successful. And at the last minute, Greenpeace and its colleagues got in touch with the Ministry of Environment, which was the sort of last step along the governmental chain that had to be addressed, and told them that this was very, very dangerous. BT eggplant was very, very dangerous. So that's on hold at the moment. So if you would like to find out more information about this whole subject, you can either go and read 1,000 articles and books in the Mann Library, or you can look at this brilliant paper, by myself, and some other people, including Frank Schottkowski, you see there. And you can get an idea of what our plan was, what the background was for this, this project, this strategy. Frank and I are working on a paper now, which will be published in GM Crops and Food, which summarizes the entire progress that was made during the 12 years. This one down below is one that I wrote with a couple of colleagues in some work we did in South Africa. I didn't want to include it here because we're pretty short of time, but I was asked to go to South Africa as a consultant and assess the impact of BT cotton, insect-resistant cotton, on the Zulu farmers of an area called KwaZulu-Natal. And this paper here summarizes what was found. What I could say is that having shown you all those graphs, global this, global that, this brings it right down to the personal level. I went there and I found that the Zulu farmers, their only cash crop was cotton. That the American bollworm was destroying their crops. Just like in the case of the eggplant, masses and masses of pesticides were being put on to save their only cash crop. I'm sorry, I should have asked you all to turn off your cell phones. Whose is that? Oh, it's mine. I'm sorry. But basically, they're in a mess. And the idea was to try and write up something about the way in which this cotton had been introduced, the importance to the farmers, and so on. I remember at one stage, I don't know how many of you know about the history of the British and the Zulus, but it wasn't all pleasant, and very difficult people there. Didn't like to be conquered by the British for some reason. I guess there's some people around here who felt the same way. So. And basically, the only amusing part of all this was they were in a circle around me in this village. They all had these farming implements, which were looking more and more like weapons to me. And they asked this Afrikaner guy from Delta and Pine, who I was with, who was shepherding me through. In their language, they said, "Is this man going to take away our special cotton?" And the Afrikaner told me afterwards, because, you know, Afrikaners and the Brits had a little bit of a scuffle called the Boer War. And he said to me afterwards, "You can't imagine what agony I was in." Here was a chance to say, yes, and get revenge. But anyway, but the fact was, it was very, very important to them. Well, let's get on to the serious business of perception again. Everything was going great. They got back their cash crop because of the BT cotton. Within a couple of years, a disaster happened. The cotton crop went, the BT cotton crop. Terrible. There was a major drought, and various other things took place so that their cotton industry collapsed again. I must say it was just a temporary thing, but you can imagine BioWatch, for example, which is closely allied to Greenpeace, etc. They wrote an article pointing out how BT cotton was no use to the Zulus, just as they had said there's no sustainability associated with this, blah, 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 blah. Then another group, luckily, wrote in Nature that, in fact, this drop in the cotton industry had nothing to do with BT cotton. It was to do with cotton prices having fallen, various nasty things happening with the South African currency, the rand, with the drought. Nothing, and they state, had nothing to do with the BT. So these are the kind of perceptions that you have to watch out for. I'm not saying that everything's perfect about GM crops. I'm not saying that there will not be problems in the future, but I am saying be careful what you believe. The ultimate story here with regard to giving a misperception, that's 5, misperception, is I was in an OECD conference in Paris, and an Englishman stood up and showed a correlation between the introduction of GM crops into Britain, before they banned them, of course, and the death rate of a certain British bird. It was a fabulous correlation, if you don't like birds. So, perfect. Well, the reporters went rushing out, started writing their papers and everything like that. They didn't stay for the question. The questions were interesting, but the best one was this. Another Englishman that happened to know about the scene in Britain, he said, excuse me, there's just one fallacy with your argument here, that GM crops kill birds. That bird is only found in the Highlands of Scotland. There is no agriculture in that particular ecosystem, in that particular part of it. Those birds don't even come across crops, let alone GM crops. Could you explain to me what the cause and effect is on it? But next day in the newspapers, headline, GM crops kill birds in England. Scotland, sorry, apologize. But can you imagine? This is the kind of thing you have to watch out for. I'm not saying that everyone who's against GM crops is wrong. There are valid arguments to be made. But make sure they're valid before you go off saying GM crops kill birds in Britain. Thank you very much.
One of the challenges for GM crops is when crops going on the world market rather than staying in this country, those being produced, which may not be a problem. But I recall that when the GM soybeans became available,
GM which, sorry?
soybean,
Oh, soybean, yeah.
that there was approval in one country and not the other between Argentina and Brazil.
Mm-hmm.
And there were farmers crossing over to get it, to bring it into
That's
the country.
right.
Wasn't allowed. And there were issues when they shipped product to Europe, which had already at that point banned crops.
Mm-hmm.
So there was some marketing problem. So that is an economic side issue with the GM crops. I don't know what the case of the eggplant in India versus Bangladesh. Is there crossover there?
There's envy by the many Indians who wanted to adopt the BT eggplant technology. And just as they're rivals in cricket, they're rivals in GM crops too. The problem is that with a Minister of Environment saying you can't do it, that was that. But there's a lot of envy there, and I think that Bangladesh is going to shoot ahead in terms of the business side of the eggplant, not only in Bangladesh, but also in the international market as well. That business in Brazil and Argentina, you know, there's tremendous rivalry between Argentina and Brazil, as many of you will know, in soccer, or what they call football. And when the Brazilians stopped commercializing GM crops, and soybean in particular, the Argentines' business in that area shot up for export and within the country and so on. And Brazilians were really upset because there's so much competitiveness between the countries. And that feeling actually had an effect on the Brazilian parliament. So, crazy stuff.
Peter, I really like, given my own research background, I love the idea of moving genetics within germplasm from one to another very effectively and very fast. I think we can certainly do that with editing. Question is, what level of safety checks do you think you have to do if we're essentially moving native genetics around? So I think if the threshold gets too high, then we'll just go back to GS. So where do you think the appropriate safety check of editing movement of germplasm
The safety in terms of what? Messing up the molecular or the genome of the one that you're transferring the genes to from wild?
I don't know, to convince the public that we didn't mess things up.
Okay.
Yeah, so I gone to the germplasm bank, I moved an allele from one there into another one. What's the threshold do you think we need to have in place to prove we haven't
Good question. I think with the same threshold as we have now, one of the first things that's done when, shall we say, a foreign gene or whatever you like to call it, the gene that you've targeted to be put into the variety that you're wanting to improve, the first thing that's done is to do a molecular analysis of the transformed or genetically engineered crop. And that's done at the minutest level, looking for any abnormalities apart from having put that gene in that may have occurred, unexpected ones, undesirable ones. So at the molecular level, you have to show that that's the only change that's occurred. So that's how it starts. Then, once you're satisfied that that is the case, then you start growing out these transformed plants and doing all kinds of checks in the greenhouse to find out whether the plant itself looks the same as the initial variety, the non-transformed variety, etc., etc., etc. So that's the answer, really, starting right from the very first analysis of what they call the event, when you put the gene into the target crop. Right from there onwards, they're looking for abnormalities. And that's done now. It won't be anything new if we go to looking at a germplasm collection, taking wild gene from a wild potato, for example, putting it into a commercial potato with exactly the same thing. You look for abnormalities from that gene. And sometimes you get them too. And those particular events are thrown out. Does that answer your question?
Yeah, I mean, I think the issue is the way you're laying it out. It's going to cost us way more than if we just high-speed GS. Okay? And so, I think high-speed genomic selection, allele back in. So if I have a regulatory threshold that's going to require me to do 4 years of evaluation and pick out minute differences, I'm better off just using genomic selection, pushing 4 generations a year in a greenhouse, and do it that way than I am. And I think a lot of us are thinking we really want to edit 1,000 things at a time, not one.
Yeah.
And the molecular
absolutely
side, no
right.
We can totally do the molecular side and show that we're only keeping the ones that we did. I have no problem with that. It's at scale, how do we cost effectively?
Well, at the beginning, I said that GM should not be used in general as a cure-all, that it should be used, in my opinion, only when other approaches are not going to work. If you have a situation where another approach will work for a particular crop and a particular trait, go with it. Even if other non-GM approaches might work, go with them. But in those cases where they will not work or are unlikely to work, look into GM.
I think the single event works, it's the 1,000 events doesn't work. And so if you set up a regulatory environment for that one event, it then precludes the application of high-scale GSIP.
The bottom line is at some point admit whether a technology itself is generally harmful or not versus the application of it. Again, is moving a protein over dangerous? Well, if it's an allergen harmful to certain people, well, yes, regardless of how you transfer it. But if the technology itself is not the issue, then they're going to be able to parse that. But one of the questions that are related to this is this off-cited question about speed. And the problem is, what is the start point? Because the early GMO work all used genes that had already been found and owned and proven to be the case, and that was their starting point. And so the time they were measuring was after years of work had already been done to get that point, and then just looked at the transfer in, and then did not consider the analysis afterwards. And so they say it's very short. And they say, well, breeding takes a long time. Well, the long time of breeding included searching for the right expression, discovery of the gene, the mapping of it, all the things that allow the gene work to be done. And then you don't have the years of regulatory testing afterwards. So has anybody actually done a true comparison? Not these, rather flippant ones that are setting up the straw man, but a true comparison of what actually speed takes. And as you're saying, with the high-speed selection, genomic selection, you can go extremely rapidly. You said 2 years?
I mean, if you just want to clean up with one gene and clean up one background, you can, and if say we're doing greenhouse tomatoes, you can get 3 generations a year, and you can clean it up extremely precisely.
2.5 years to process a tomato, a fresh market tomato, and have a trial, a field trial. I mean, that's all it took. So yeah. So how slow is breeding? Processing tomato, the fresh market tomato is totally. So it gets the question.
I can tell you definitively, but I don't know.
This has been a production of Cornell University, on the web at cornell.edu.